Some of this is going to be new, not necessarily new information, but new in the context of this chapter. I wanted to talk about the stages first of dendritic cell, and then talk about the role that that plays in terms of activating T cells. So for dendritic cells, the stages that it has is there's the immature stage and then the mature stage. Fine that we're talking about dendritic cells here. And then as it's going from the immature to the mature stage, I just wanted to briefly mention that it's migration process. So we have migration taking place. So in the immature stage, where is it located? Well, it is located in, you can think of it as the infection site, or I just like to think of it because it's smaller and is less, for me to write, it would be at the site of the tissue. Whereas once it becomes mature, it's located at the secondary lymphoid. Okay, so what guides this movement from the tissue to the lymphoid organs? Well, that's the process of migration, which is regulated by CCR7, so the receptor 7, on the dendritic cell, okay, and then the CCL21. So this is not only controlling the migration, but this is also controlling the development. The dendritic cell is going to go from being this immature, globular type of a structure to being this mature, highly dendritic type of a structure. But yeah, like I said earlier, this tends to have a globular type of a morphology to it, whereas the mature dendritic cell has a dendritic. And I'm a huge fan of the whole structure determines function things. So we're about to find out what the difference is between these globular and then dendritic structures, and then talk about the functions. The main thing that the immature dendritic cell is doing is environmental sampling. And there's two mechanisms that it has to do this. The first one is called micropenocytosis, and then the other one is macropenocytosis. Okay, so as their name both implies, micro is small, macro is large. And what I'm about to tell you about these is obviously, we're all about T-cell mediated immunity, so in this context, we're definitely going to be talking about antigen presentations and getting those antigens. But I want you to think about micropenocytosis versus macropenocytosis, and that one of these is receptor mediated and the other isn't, at least in this context. Be thinking about which one of those you think it is, but for now, let's just talk about also mentioning that both of these guys, we're talking about an endosomal pathway slash MHC type 2. Which one of these do you think is receptor mediated? Well, it's the micropenocytosis, which is receptor, because we're dealing with such a small volume, we want to be sure that we're being specific, whereas macro, such a large volume, we don't need to be because it's arguably somewhere. And the only thing that I wanted to mention, one last thing with these dendritic structures here, is that this is going to increase the surface area of exposure for the major histocompatibility type 2. And they're going to have a lot more MHCs. So we've increased our surface area and we've increased our gene expression for MHC type 2 because in this, whereas with the immature we are just all about environmental sampling, for the mature dendritic cells, these are all about lymphocyte activity regulation, lymphocyte activity control. And the two processes that are involved in this, hopefully I can fit them both on here, is T-cell migration and T-cell activation. Probably not going to have room to list the in-depth details of each and every one of these, but let's just talk about T-cell migration first, if we can make it fit on here. Involved in the roles that they play is that they have ICAMs. In this context, I am talking about, whoa, green, or blue. I don't know what color this is. I'm colorblind. ICAM 1 and ICAM 2. These two bad boys here are going to be interacting with, and I will do it on the magenta cell surface, because if I say T cell receptor, that almost makes me think I'm not talking about the whole alpha beta thing there. But anyways, the ICAMs of the mature dendritic cells are going to be interacting with the LFA1, which hopefully that makes sense to you. They're both integrins. Weird enough, they also understand that anytime you have an LFA that's going to be interacting with some type of a ICAM, so the dendritic LFA is going to interact with the T-cell ICAM3. To see if I can closely mention this, I'm going to have to probably stretch this out and make this map look really, really ugly, but another type of a compound that exists with the ICAM3 is known as DC sign. This is also going to be interacting with the ICAM3 on the T-cell surface. Really just three things that I want to say about DC sign in terms of its structures is that it is a lectin. It's an adhesin, only present. Very last thing, and let's really stretch this out. I know it's getting really hideous and ugly when we're talking about T-cell migration and dendritic cells. Really stretching this out. Really ugly, hideous concept map. But the last thing that I wanted to talk about would be LFA3 on the dendritic cells and its interaction that it has with CD2 on the T cells. So kind of like when we talked about the whole interactions between there, but the binding of LFA with CD2 is going to result and a conformational change kind of across the board that's going to signal certain events that are going to cause the actual interactions between other adhesin and integrin molecules to get strong. This results in a conformational change that gives us stronger bonds for everyone. And then the last thing that I wanted to mention on the subject of T cell activation, I know I don't have a lot of room here, but, and I'll do it in, I guess I'll just stick to it in white, is that this results in something called the supramolecular activation complex / the synapse. And we'll talk about that later, and that is also happening between those two things there, between themselves and that of the T cells. So let's move on to the next slide. For dendritic cells, the next thing I wanted to mention was their ability and the roles that they can play in processing and in presentation, which is kind of review, but not necessarily, we haven't gone in this much depth with specific cells. So there's really just two things that I wanted to say here, is that in terms of if they actually do get virally infected, they have some pretty nifty mechanisms that they have that actually benefit them, which is why I think there's very few viruses, at least that I can think of, I'm not a virologist, that actually will actively and isolatingly speaking only infect dendritic cells. And I just wanted to briefly review the roles that they have in terms of their toll-like receptors. So here's the interesting thing. If they are virally infected, regardless of where they are, and I don't quite understand the in-depth mechanisms with this, I assume it has to do not necessarily solely with the whole migration forces and things like that, but with a kind of an intrinsic alarms going off inside. But if they're virally infected, they will migrate themselves to the secondary lymphoid tissue. And hopefully if they're here, they'll be in a high density areas of CD8 cytotoxic T cells that will be able to interact with their MHC type 1 molecules. Or MHC type 1 proteins. Everything's a molecule, but protein would be more specific. But let's say that it doesn't make it all the way to interact with this, that it dies before it can do that. Then what will happen will, oh my, I did not mean to do that. I'll just write it in white. It will virally infect, it'll put itself into the close enough proximity to where it will virally infect other dendritic cells. I'm just going to say DCs for that. And then from this processes here, we can either get the presentation of self-peptides through the MHC, or we can actually undergo cross-presentation happening there. Obviously, this is all about the MHC type 1. But these guys are extremely efficient at antigen presentation, be it MHC type 1 or MHC type 2. And we'll talk about later why they're just so damn good at that. Next thing that I wanted to mention with regards to toll-like receptors, and this will help you understand a little bit about their function, is that these guys do not have TLR9. None of that going on. However, it's the actual toll-like receptors that play a role in actually activating the dendritic cells. Activates the dendritic cells. I know we had also mentioned about the chemokines that play a role in this, but majority, the whole changing that they have in terms of their morphology, that's really an intrinsic internal alarm thing is what I like to think about it, or an internal change happening more so than something that's driven by external factors. So once we have TLR activation, we're going to start to increase our MHC type 2 expression, kind of what we had saw in the PowerPoint slides there with the changing colors. We're going to start to become more sensitive to things that would trigger a phagocytic event or environmental sampling, if you could think about it. They're going to start to change patterns in gene expression, which is going to give them a dendritic morphology, which is good if you want to increase the amount of surface area and the amount of exposure we have of things to that MHC type 2. And lastly, and certainly but not the least, when we start to talk about the T cell activation and when we mentioned the co-stimulatory signal, it's the B7 that we can find on the dendritic cell that arises in response to activation of the toll-like receptors that enables these things to start moving and to be able to start being activated. Siren has stopped. Let's review. So for number one, think of this question. It says that when an immature dendritic cell becomes an activated dendritic cell, all the following changes occur except what? Do you think that it's A, confinement to T cell regions of the lymph node, B, development of dendrites, C, increased MHC2 expression, D, loss of antigen processing functions, or E, expression of TL9? So pause the video if you need to and wait for the answer. All right, so the answer to that is E, expression of TL9. Remember I said that they do not express. That's the one toll-like receptors that you're not going to find inside of MHC, of dendritic cells.